OK. Now let me get to the second paper that I showed you and this idea of genetical genomics. I think this is a really interesting paper because it's one of the few that I've come across where both gene expression profiling and genotyping, or at least the promise of genotyping, are kind of fused in this new field of electronic QTL or genetical genotyping, genetical genomics. Let me just walk through this figure. In this paper, what they did is they took cell lines from, I guess, 33 individuals, and they did microarray analysis on them. So these people are all different. Some of them were twins, and some of them were unrelated individuals. And what they wanted to know is, how do the expression patterns of genes in these cell lines from people compare between those individuals? I wanted to ask a couple questions. One, are there some genes that are more variable between people than others? And then they wanted to also ask, are siblings and twins more similar in their expression profiles than unrelated individuals? So here in this first slide, you can show, they did a nice thing. You know how I mentioned that there's a lot of variance in the assay itself. It's kind of a noisy assay. And in fact, depending upon the intensity of the signals in that plot I showed before, low intensity signals tend to be more variable. So the way that they showed the data, and this is kind of standard in genomic analysis, that you get kind of schematic ways that allow you to kind of take all the data in at once. This represents the variance, in other words, the amount of difference in the measurement of a given gene between replicates, because what they did is they measured each person twice, versus the variation between people. And what you can see is that there's more variation between people than there is between replicates. That's good. Their technique was good. But then they said, well, OK, we really don't want to be looking at genes that are different between people, or that we think are different between people, just because they happen to be variable genes. Hard to assay, low intensity, hence quite variable. So they said, let's plot one against the other. Let's just take this edge of the data. Because this edge of the data represents the genes that are tightest in replicate measurements. In other words, when you do the analysis twice on one person, they don't vary much, but are most variable between people. In fact, this gene, this is the most variable between people, but it's also slightly more variable in terms of analysis. They said, we really don't want to throw out any data that's just intrinsically variable. And so let's look at these genes. And so they came up with a number of genes that they could pursue further. And here are 5 of them. These are just the human genome names for the genes. And you can see that we're talking about a 1, 2, 3, a 4-fold variation. These genes are expressed 10,000 times higher in some individuals than other individuals. And here are others. OK, what this suggests is that these are good candidates for genes whose expression results in differences between people. Because a gene that's expressed at the same level in everybody is unlikely to be resulting in differences. Now, I should point out, the genes could be different in terms of their structure. This doesn't ask anything about the structure of these genes. It just says how much of the gene is expressed, what the transcript level is. The actual form that the protein that's encoded by the gene takes could be different. And they can't measure that kind of variation. But in any case, what's really intriguing is that they then said, OK, how about the variation between these 5 genes in unrelated individuals, siblings, and twins? And what you can see is the twins are nowhere near as variable as siblings, and siblings are not as variable as unrelated people. And that makes sense. And this gets back to the genetics we started with. These siblings are clones of each other. They're identical. These are identical twins. In other words, this difference is not random. It's due to genetic factors. And those genetic factors are probably, among other things, SNPs, or other variations in the sequence. So you've now just used gene expression to identify genes that might vary. And what the term genetical genomics refers to is the fact that you actually might just use this gene expression as the trait that you study. You don't even have to worry about phenotype, because phenotype can be a difficult thing to measure a lot of the time. Here, your phenotype is just the amount of a transcript you have. And you can map it to genetic loci, to genetic markers in the same person, thereby kind of cutting out the human being entirely. But what I mean is that you can actually study this in an in vitro situation and probably get a lot of information. And whereas, let's say, for instance, in Esteban's studies, they look at asthma, the phenotype is an extremely expensive and difficult thing. And the whole value of what Esteban's done is he's got patient data on these people. Well, you can only look at a trait that maybe involves a handful of genes at a time that way. This is an alternative complementary approach where you can't pick what genes and what traits you're looking at, but you're looking at 30,000 of them at once. So it's quite powerful. Parting thoughts. This gets the omics part again. OK. When I was writing a talk a few months ago, I thought omics, and I remembered that I was an ecology major, that both ecology and economics have as their root the term oikos, which is Greek for house. I'm not quite sure why house, but both ecosystems and economic systems have always been understood to be complex webs of interactions. And that's always been known in biology too, but the second half of the 20th century was characterized by an extremely productive reductionistic paradigm, when it made a lot of sense to actually forget that for half a century. And I mean, the inroads that were made in molecular genetics by basically physicists who came into biology were made because they said, you know what, you biologists are just systematizing everything. You're just coming up with names. You're not actually dissecting it. And they dissected it. What's interesting is we've kind of come full circle to the point where now omics is back. And what's interesting is there is, in fact, a journal. I didn't make this up, but it's called Omics, a journal of integrative biology. That's really what omics means. The problem with this is a lot of people won't be happy with the idea of systems biology, of knowing everything, because they say you're just measuring. People have become so happy with this idea of hypothesis-driven reductionistic science that actually going back to what's really biology's roots can sometimes appear somewhat odd. And you don't really need to have a systems biological approach where you measure and characterize everything. But I argue that we need to do systems biology. And let me give an example with Vioxx. Everybody knows that a few months ago, Vioxx and now Celebrex and a lot of other drugs have been taken off the market. Now, the interesting thing about these drugs is they were designed. They were some of the first generation of drugs that were designed with hypothesis-based models in mind. People designed them to not cause problems, to get around the problems that were known to be caused by things like aspirin and non-steroidal anti-inflammatory drugs. There were two enzymes known, and they designed these to only hit one. But they all start to cause heart attacks, which is paradoxical. It's one of the things they're not supposed to do. Aspirin decreases heart attack risk. And what this shows is that you really have to know everything ultimately. And that doesn't just mean everything in one individual. It kind of means you have to know everything in everybody. And I mean, if that sounds kind of hopeless, I would kind of fall back on, well, we don't have to know this stuff, right? Because right now people already live a relatively good lifespan. Scaremongering is sometimes silly. We don't have to know this. But the point is that in the kind of 100 to 200 year time frame of where biology is going, this is where we're getting into, kind of personalized medicine or personalized biology, understanding that variation exists out there and that to capture it is going to require a degree of data capture and analysis that's really not common at this point, but probably will become more common over time. And I thought this was funny. Finally, this even applies to the information itself. I'm sure there's not a practicing biological researcher out there right now who does not feel completely and utterly overwhelmed by the flood of information. It's just not even it's not capable to keep up with the literature, and people even said we need to start to apply kind of high throughput techniques to reading the literature that we're all producing to be able to encapsulate it in ways that a human can understand. So these are just pictures to show you that these are the kind of maps that exist of metabolism, and they now need to be described at a genetic level. Let me skip over this. And end with this. I do think the future of biomedical research then, if this kind of paradigm of high throughput approach is going to occur, needs to be modeled more on something like the way NASA or the National Oceanic and Atmospheric Administration work, which is that they have large teams of people who work together, because just speaking personally, it's all I can do to keep the technology in hand, and that's why I work with people like Esteban, because I don't know what he knows. He doesn't know what I know. Together, we can actually do these analyses. You can't do it yourself anymore. And with that, I want to just thank the various groups with which I'm associated, and notably Esteban Burchard and the National Center for Minority Health Disparities, who've supported a lot of this work.